Today, we're diving head first into a microscopic war zone. It's a place where survival hinges on one of the most brilliant, most resilient pieces of engineering in the entire natural world, the bacterial fortress.
So, let me start with a question for you. Picture the pressure inside a car tire, fully inflated. Now multiply that pressure by 20. What tiny, single-celled organism do you think lives its entire life under that kind of constant, crushing force from the inside out? You got it. The answer, as wild as it sounds, is a single bacterium. That tiny little life form is packed with so much stuff that the pressure inside is constantly trying to make it explode.
So, how on earth does it survive? What's its secret? Well, it's a perfectly engineered suit of molecular armor, what we call the cell wall. And believe me, this isn't just some flimsy bag. It's a high-tech, life-saving fortress.
So in this explainer, we're going to take a full tour of this incredible structure. First, we'll get a grip on the insane pressure problem bacteria are up against. Then, we'll check out the two main architectural blueprints they use and the molecular assembly line that builds the whole thing. After that, we'll see how our best weapons, antibiotics, are designed to bring that fortress crashing down. We'll even look at some of the bacteria's own advanced weaponry before we wrap up with the state of this microscopic war today.
All right, let's start with the fundamental problem here. You can think of a bacterium as this really salty little bag floating in relatively fresh water. And as you know, nature loves balance. So water just floods into the cell trying to dilute it. This creates this immense outward force called osmotic pressure. And without a super strong wall, that bacterium would just pop instantly, like an overfilled water balloon.
And the scale of this force is just staggering. For some bacteria, it's as high as 20 atmospheres of pressure. That's why this rigid exoskeleton, which scientists call the sacculus, isn't just a nice-to-have, it is absolutely essential for life. Its strength is literally the only thing standing between the bacterium and total annihilation.
Now here's where it gets really interesting. Not all bacterial armor is built the same. In fact, there are two main architectural blueprints. And the difference between them is so fundamental, it's the basis for one of the most important diagnostic tests in all of microbiology, the Gram stain. On the left, you've got the gram-positive design. Think of it like a heavily armored fortress, a single membrane protected by this massively thick wall designed for pure, raw defense. On the right, you have the gram-negative citadel. It's a more complex, more strategic design, with a thinner wall that's protected by a special outer membrane that acts like a selective shield. Let's dig into what that really means.
So the gram-positive fortress is all about brute force. Its wall is this thick, dense mesh of a material called peptidoglycan. You can just picture it as molecular chainmail, layered over and over again for strength. This super thick wall is what traps the purple dye in a gram stain test, and it's the design used by famous bacteria like Staphylococcus and Streptococcus. The gram-negative citadel, on the other hand, is way more sophisticated. It has a much thinner layer of that peptidoglycan chainmail, but, and this is the key, it's protected by a second, outer membrane. This outer layer is a brilliant defense. It acts like a selective bouncer at a club, keeping out large, dangerous molecules, including some of our most powerful antibiotics. This is the style you see in bugs like E. coli and Salmonella.
Okay, so we've seen the final structures, but how in the world are these incredible fortresses actually built? It's this fascinating, non-stop process that's just like a molecular assembly line, and it happens in 3 distinct stages.
It's really a beautiful 3-step process. First, deep inside the cell, in the cytoplasm, all the little parts for a single building block are carefully manufactured and put together. Second, a special lipid shuttle grabs this finished block and ferries it across the cell membrane to the outside. And third, on the exterior, a team of enzymes that we call molecular masons grab these blocks, link them into long chains, and then stitch all those chains together to create the strong, cross-linked mesh of the wall.
And this brings us to probably the single most important component in this whole operation, a molecule called lipid 2. This is it. This is the complete, ready-to-install master brick, already attached to its delivery shuttle. And here's the kicker. The cell only has a limited number of these shuttles. So if you can take lipid 2 out of commission, it's like seizing all the delivery trucks at a construction site. The whole project grinds to a halt. And for a growing bacterium, that is a death sentence.
And that, of course, brings us to the siege. For almost a century now, humanity's greatest weapons against bacteria haven't been bombs or bullets.
They've been tiny molecules designed to do one thing and one thing only: sabotage this critical construction process. It's a brilliant strategy, really, because our own cells don't have these walls, which makes it a perfect target.
This quote just nails it. The single most vulnerable part of the whole process, the real Achilles heel, is that very last step. It's called transpeptidation, which is just the fancy term for when those molecular masons stitch the wall together for strength. And that's exactly where our most famous antibiotics strike.
And there are two classic ways to pull off this sabotage. Penicillin and its relatives, they're tricksters. They look almost exactly like the building blocks, so they fool the molecular masons into grabbing them. But once a mason grabs a penicillin molecule, it gets permanently jammed. It's basically poisoned. Vancomycin uses a totally different tactic. It ignores the worker and goes straight for the materials. It latches onto the lipid 2 building block and forms this bulky cap, making it physically impossible for the mason to even use the brick. Either way, construction stops. The wall gets weak, and that internal pressure does the rest. Boom.
But hold on, these fortresses are not just for defense. Oh no. Many of them are armed to the teeth with these incredible molecular machines, actual weapons designed to attack our own cells and even rival bacteria.
I mean, just check out a few examples of this stuff. The type 3 secretion system is basically a molecular syringe that bacteria use to inject toxic proteins right into our cells, hijacking them from the inside. The type 4 system could do that too, but it can also inject DNA, essentially reprogramming other cells. And then, and this is my favorite, there's the type 6 secretion system. It is, and I'm not making this up, literally a nano crossbow. It's a spring-loaded weapon that fires a toxic-tipped spike to puncture and kill any cell it touches. Just incredible.
And all of this brings us right to the modern battlefield. You see, for billions of years, bacteria have been slowly upgrading their defenses. But in the last 80 years or so, our widespread use of antibiotics has put that evolution into hyperdrive, leading directly to the crisis we now call antibiotic resistance.
And bacteria have evolved some absolutely brilliant counter-strategies in this arms race. Some of them engage in enzymatic warfare, producing these little enzymes that hunt down and destroy penicillin before it even gets close to the construction site. Others, like the infamous MRSA, just modify the target. They bring in a new kind of molecular mason that penicillin can't recognize or bind to. And some, like VRE, completely redesign their building blocks, changing the chemical signature just enough so that vancomycin can't grab onto it anymore.
Which leaves us with a huge and, frankly, pretty critical question for our time. Bacteria have been perfecting this molecular armor for billions of years. We've only been seriously trying to breach it for less than a century. As they continue to adapt, to evolve, and to outsmart our very best weapons, you really have to wonder, in this ancient, ongoing war, can our science actually keep up?